Hello everyone, welcome to Deepti's biology class. We have covered the first event of post-fertilization, that is endosperm development, and this is the second event of post-fertilization event, that is embryogeny. Embryogeny is the formation of embryo from zygote. How the zygote is developing into embryo, that is what we call embryogeny or embryogenesis. Now, based on in which type of plant this particular process of embryogeny occurs, it is classified into two. One is dicot embryogeny and the second one is monocot embryogeny. So let's start with dicot embryogeny with the help of this particular diagram. So keep your notebook with you so that you can draw each and every stage which is given here. So the zygote, which is diploid in nature, just we are writing here the micropylar as well as chelacal end. So this is the micropylar end and this one is the chelacal end. Now the zygote undergo mitotic division. When it is mitotic division, whatever new cells which are formed here is going to be diploid in nature, that is 2n. Now don't get confused by seeing some other textbook. Maybe you see that this diagram is opposite. This portion may come downside or this portion may come upward. You have to just see the micropylar as well as the chelacal end only. We do not use the term upper or lower, but with reference to this chelacal or micropylar end, we will be seeing how many types of cells are formed. Now what happened now? The zygote undergo mitotic division to form two cells. Now the cell which towards the micropylar end is termed as basal cell and the cell which is towards the chelacal end is termed as terminal cell. So basal cell as well as the terminal cell. Which one is basal cell? The cell which is there in the micropylar end. So that you can say that there is a horizontal division. What is horizontal division? A cell which is, this is the zygote, which divides into two. In this axis it divides. So there will be two cells which are formed. This one is termed as the basal cell and this one is termed as the terminal cell. Right? This is what happened now. Now what happens is that each cell undergo division. Basal cell as well as terminal cell. Both these cells undergo division to form new group of cell. When the terminal cell undergo division, it forms a mass of cells which are 4 in number. How this will be there? Suppose this is this way there is, right? Two cells are formed. One is terminal and one is basal. Now this terminal cell, particular cell, what happens? There will be one division followed by next division. So what we will get? 4 cells will be there, right? The same happened here. Now what happened here is that terminal cell undergo 2 division which forms 4 cells, right? This part where the 4 cells are formed, we call it as pro-embryo. What do we call this one? This is what we call pro-embryo. Now what happens to the basal cell? Basal cell undergo a repeated division. Like suppose this is a basal cell, one basal cell which divide into 2. Each will divide now. 4 will be there. Now all the 4 will divide. This way the division will happen and as a result we will get a set of cells or a chain of cells. We call this chain of cell as suspensor. And the last cell of the suspensor is just like a round or oval shape and we call it as hostorium. So what happened? From the terminal cell, pro-embryo is formed. From the basal cell, suspensor, chain of cells which are formed, and the end, we can say that the end or else the cell towards the micropylar end is little swollen or else we can say it is round in shape. We call it as hostorium. Now what happens in the next stage? Each cell of this particular pro-embryo divides. So what we will get? We will get 8 cells. We call it as octant. Octant means 8 cells are there. So suppose if this is that cell stage where 4 were there, right? Now each cell undergo division. So what we will get? We will get this type of structure, right? So 4 cells were there. All 4 undergo formation of 2 new daughter cells and we will get 8-celled stage in the pro-embryo. That is the first stage which happened here. And the second change which happened is that the cell of the suspensor this out of the cells of the suspensor, the one which is just below this pro-embryo, this particular cell, you can see I have changed the color here, right? This cell, now we call it as hypophysis. So what is hypophysis? Listen carefully. Hypophysis is the cell which is just below the proembryo, or else we can say the cell of the suspensor towards this particular proembryo is termed as hypophysis. So the same hostorium is here and the suspensor is also here. What change happened from this stage to this stage? In proembryo there were 4 cells, is converted into or divided to form 8-celled stage. One cell of the suspensor just below the proembryo. Proembryo meaning is that just before the stage of the embryo formation, become hypophysis. We will see what happens to this hypophysis and all later, right? Now, this hypophysis is very important also, and remaining structure is just as it is. Now, till now mitotic symbol division were there. Now these undergo a kind of division, we call it as, just you note down, I will rub it, okay, that is periclinal division. Where happened? From this stage to this stage, which type of division happened? Periclinal division. I will show you just now how the periclinal division will be there. There was the proembryo, right? This proembryo which I am drawing here. Now what happened? First it had 4 cell. Later it divided into an 8-celled stage, right? These all are the 8 cell. Now when there is periclinal division, what happens? This way it will divide. One cell will divide to form 2 cell. This way each cell of that octant is divided to form more number of cells. So what we will get? An inner mass of cell and an outer mass of cell. That is what happened in this particular stage. So proembryo undergo periclinal division. As a result, we will get a mass of cell. Next, what change happens? From what is the difference from this particular diagram to this one? So first one, what happened? This proembryo becomes again increased mass of cells, right? Increased mass of cells are there now. Now apart from that, this region, that is the hypophysis, is going to divide number of times, and we will say 8 cells which are formed there. So this particular, which we have drawn in purple color here, undergo division, and we will get a mass of cell here. And the suspensor, hostorium, everything remain same. So here we will see what is the fate of all these structures. Fate means what happens to all these parts in future. Let's see what is going to form from all these structures. Now, what is the function of this much structure? What is this much structure? This is actually the suspensor as well as the hostorium. That is suspensor as well as the hostorium. What is the function? We can write this way. Please note down. Later I will rub it. The function of this one is to push embryo into endosperm. Push embryo into endosperm. So let's see what is the meaning of the sentence which I have written just now. We know that this is our ovule, right? Embryo sac is there. In this particular embryo sac, we know where is the zygote. This is the zygote, right? And what will be here? There was this endosperm, and we have studied the endosperm development, right? This entire region endosperm develops. What is this endosperm? This endosperm is the food for this growing zygote now. After the endosperm formation, entire embryo sac is filled with mass of this endosperm tissue. Now, with the help of this hostorium, this embryo is pushed into this one. Why it is pushed? So that all the food can be all the food can be utilized. So this particular zygote will be pushed, or the growing proembryo, this entire structure, we can say this proembryo or this structure is pushed into the endosperm so that it can use these nutrients which are there. So that is the function of this long chain of cell. What is the function? to push the embryo into the endosperm so that it can utilize the entire available food. So we know the function of the suspensor now, right? To push this entire structure into the endosperm so that this can utilize that food. Now next structure is, what is this we call it as? This is what the hypophysis. This is the name of the structure. Now what is the fate of this hypophysis? The lower, so we can see two half, right? One half here and another half here. The lower half towards this micropylar end is going to become the root cap or root tip. What about this half which is towards the proembryo? This is going to be the root cortex. If you have studied the structure, internal structure of root or anatomy last year, you know there is root cortex is there, epidermis is there, all these structures are there, right? Now, so out of this hypophysis, the half towards the micropylar area is going to become root cap or root tip, and the above half, or else we can say the half which is towards the hypophyseal half which is towards the proembryo is going to become the root cortex. Now what about this proembryo? One half of the proembryo is going to become the hypocotyle. We will study what is this hypocotyle and all. And next half is going to become two structures, that is the plumule plus cotyledon. Hope you have noted down this one. Now please understand this one. From what is the fate of the proembryo? If you divide this way, one half is going to become plumule. Plumule is actually the future shoot portion. What can you conclude from this one? Hypophysis, half of hypophysis is going to become root cortex, remaining is going to become root cap. As a result, we can say this hypophysis is the future radicle. What is radicle? The future root. That means this much area is going to become the root in future. What about this proembryo? The lower half is going to become, this much is going to become the hypocotyle region, right? We'll see what is that. Now what about this one? Upper half or the part which towards the chalazal end is going to become plumule as well as the cotyledon. This should be your reference. This image is actually your reference, this entire image, because here we have written what is the fate of all these things, right? Now let me rub all these things. Hope you have noted down. Then only we can go to the further one, right? So we know the fate of this entire structure. And remember one more thing, this stage is termed as globular stage. Why? Because the proembryo looks like a globular mass of structure. Based on the shape of this particular proembryo, this is divided, this phase is termed as globular stage. Now in the next stage, what is going to happen is that, so there are multiple stages in between, we are just avoiding all those things and we directly come to another stage. We are not doing anything about the suspensor, because the function of the suspensor is only to push the remaining part, that is hypophysis and proembryo, into the endosperm. So we are not going to talk about this entire suspensor. What about this hypophysis? Hypophysis keep dividing and we will get an entire mass of cell. Now this first half, what was the function of this first half or fate? It become hypocotyle. That is this much. In this diagram, let me use another color for you to understand. This much portion is actually this much of this particular diagram. So that is also going to become a mass of cell, and the remaining area of this proembryo, which is supposed to be the plumule, is going to develop into this much, and this structure now start calling as cotyledon. And plumule is going to be here. We said now this upper half is going to become the cotyledon as well as the plumule. This plumule is going to become the future shoot or the stem system, we can say. Now please note down this particular stage. This stage is termed as heart-shaped stage. Why? Now this pro-embryo looks like a heart shape, right? The pro-embryo looks like heart shape. So just you have to do one thing only. Keep the reference of this particular globular diagram with the entire labeling and fate. Then you start connecting, because we are not learning the intermediate stages. So after globular, there will be heart-shaped stage. Why? The cotyledon, or we can say the pro-embryo, looks like heart-shaped. Now, after some time, what happens? Inside the embryo sac, there is limited space, right? Now, if the growth keeps happening, what happens? Since the space is not there, they start to grow. The cotyledon keeps growing, but it start bending. So we will get a shape like this. It starts bending. This, what are these structures actually? This is actually the growing cotyledon. This is actually the cotyledon which got bent now. Now this stage is termed as horseshoe shaped. So you have to remember one thing. You must remember the stages, right? First there was globular stage, then there was heart shaped stage, then horseshoe shaped. Now again, after so many division, growth, at last what are we going to get is the diagram which we have drawn here. So hope you have written up to here. So let me write after cleaning this entire area. Now these structures which are here is the, okay, let me use another color. This structure is termed as cotyledon. Now there is two tip. One tip is here and another tip is here, and this long stalk like structure we call it as embryonal axis. What is embryonal axis? And these leaf like structures which are here is the cotyledon. Now few more things are there. This tip region on this embryonal axis, and there is a lower tip on this embryonal axis. This tip is termed as plumule. What is the fate of plumule? In future, the plumule is going to be the shoot. And what about this one? This structure is termed as radicle. Radicle is going to be the, so we can write here, plumule is going to become the shoot in future, and this radicle is going to become root. Now you can see a portion which is between this area and this area. This is the point where the cotyledon attach, right? We call it as embryonal node, N-O-D-E, node. So what is embryonal node? Embryonal node is that point to which these cotyledons are attached on this entire embryonal axis. So what is the difference between embryonal axis and embryonal node? Embryonal axis consists of plumule, radicle, and this embryonal node. This entire stalk-like structure is the embryonal axis. What is embryonal node? Embryonal node is this particular point to which these cotyledons are attached. Now, let me clean this entire area so that we can once again go through that entire structure. Now please understand, I am not repeating this particular thing again. If you have any doubt, you have to repeat and watch this video. Now, this is the embryonal axis, this entire structure. What are the things made up of? The top is called as plumule and the lowermost portion is the radicle. Right? Now, there is a point to which these cotyledons are attached. This point is termed as embryonal node. So what is this entire structure is termed as from here to here? That is embryonal axis. Now what is this structure? This is the cotyledon. Now this radicle is going to become the root in future and plumule is going to become the shoot in future. Now this area between this radicle and the embryonal node is termed as hypocotyle, and this area is termed as epicotyle. Right. So this is what the structure of a dicot embryo. which consists of the completely developed dicot embryo consists of an embryonal axis, embryonal node, cotyledons are there, hypocotyl is there, epicotyl is there, plumule and radicle. If you have any doubt, please repeatedly watch this particular video. Again I am reminding you, just will go through the points. Now dicot embryogeny. Zygote divide into two unequal cells, larger suspensor cells towards the micropyle and the smaller embryo cell towards the antipodal region. Now the suspensor cell undergo transverse division to form suspensors, right? These cells, or else we have seen there was terminal cell as well as the basal cell. You can use that reference also. Basal cell will form suspensor, terminal cell will form the proembryo, right? Now the first cell of the suspensor is called haustorium and the last cell towards it is called hypophysis. If you remember that divided into chain of cell, this last, we say last, right? But according to this diagram, this micropyle, if we are taking the reference, this becomes the first cell, right? This is called haustorium and here will be the proembryo, and this cell is called hypophysis and it forms the radicle in future, because we have seen that the first half or one half of the hypophysis become root tip, another half is going to become root cortex. Whatever it is, root, that entire structure we call it as radicle. Now embryo cells divide twice, vertically once, transversely to produce two tiered 8-celled called embryo. We call it as proembryo, right? You can make your own notes also using the diagram. Now the terminal tier forms two cotyledons and plumule, the hypobasal cell which produces hypocotyl. This area you have to use using the reference of our diagram, right? Epibasal and hypo, which what we are talking about is this. Do you remember there were 8-cell periclinical division was there, right? Periclinical division was there. This if you divide into two half, right? This is what we call hypobasal. That means the below one. This is hypobasal region and this is going to be epibasal. Don't get confused with the epicotyl and hypocotyl. That was towards the end stage, right? Once the cotyledon was completely formed. Hypobasal and epibasal. What happens to this half? So here will be this hypophysis and entire structure is there, right? This hypobasal cell is going to become the hypocotyl and epibasal cell is going to form cotyledon and the plumule. Cotyledon and the plumule. Now, initially it is globular, then become heart shaped later, then finally it occupy that typical shape, whatever the shape is, right? We know that different shapes are there. Now, a typical dicotyledonous embryo consists of an embryonal axis and two cotyledon. The part of the embryonal axis above the level of cotyledon is called epicotyl. It ends with the stem tip called the plumule. The plumule give rise to the future shoot. And the part below the level of cotyledon is called hypocotyl. It terminates in the root tip called radicle. Radicle give rise to the future root and the root tip is covered with the root cap. Even we clearly know now which part become root tip and which part become the root tip or which is covered with the root cap, and we know which part is going to give rise to the root cortex also. Now some different cases are given here. Somewhere what happens, even the cotyledon also curve as it emerged, and somewhere what happens, in some cases there is no differentiation into plumule, cotyledon and radicle in case of orchids. So this much about the dicot embryogeny. If you have any doubt, thoroughly you watch it and make sure that you make the notes when you watch the video, otherwise it will be difficult to understand. Revise it so that it will be clear. Thank you for watching.